Excellent. All right. So we turn on some lights and we'll get started. We're going to be finishing up our prokaryotes today, and then we're going to be starting on our eukaryotes, which are going to be organisms that are like ourselves as far as cellular structure goes. So let's do a quick review to make sure that we remember what we've learned so far. So first of all, the proteobacteria, are they gram-positive or gram-negatives? Gram-negatives, excellent. So proteobacteria are always going to be gram-negatives. We said there's a few major groups. The first one was the alphas. What did the alphas like in their environment? Do they like a lot of nutrients, or do they have an ability to live with low nutrients? Low nutrients, excellent. And then after the alphas, we had our betas. Did they like a lot of nutrients, or do they like low nutrients? See, nodding is done. Yeah, they like a lot of nutrients, right? These are the ones that a lot of times were parasitic towards us, where they would have to use us to give them energy, ATP, things along those lines. We then said that we had the gamma group, and we said the gamma group fell into a region where we experience them a lot, right? So what examples of bacteria just in general would fall in the gamma group that are related to us? Like in our bodies? Yes. Yes, exactly. E. coli, our normal flora. Most of our normal flora that lives in our gut is going to be a part of that gamma group. Okay, so most of our gut flora. We do have a few pathogens in there as well, but for the most part, most of them are going to be ones that are going to be living in kind of a balance with us. Okay, so E. coli, Klebsiella, these are ones that can be in balance. Now, we said that a lot of these, though, were opportunistic pathogens. That was a term that we've used before. What does an opportunistic pathogen mean? Yeah.
If it goes to like another region of the body where it's not.
Excellent. Excellent. So if it's where it should be, for example, if our normal flora are in our guts, in our intestines, then we are safe. But if it goes to another region of the body that should be sterile, think of like the bladder or somewhere else, then it can cause an infection. So they're opportunistic because they can take advantage of a situation and cause an illness. They don't normally, but they can take advantage. So that gets us through to our next group, which are the deltas. The delta is a really interesting little group. There's not a lot of members in here. We said the gamma was the biggest, right? We said the gamma is where our normal flora live that have the biggest grouping of organisms. The delta is probably one of the smaller ones. These are bacteria that survive by preying on other bacteria. They're almost like the vampires. Oh, it's like perfect timing for Halloween, right? They're like the vampires of bacteria. So what they're going to do is they live in the environment, and if they encounter another bacterium, they kind of act like a torpedo. They slam into the side of that bacterium, and they hit it with so much force that it actually causes the cell to burst. It's like hitting it with a bullet. And when that happens, these little organisms immediately start sucking up all the ATP and the nutrients that were released by the cell that they sunk, so to speak. It's kind of like I said, like a little torpedo in a submarine. So there's a couple of organisms in here. One of them that's the most common is the Vibrio. And when you hear Vibrio, what shape do you think of when you hear Vibrio? Yeah, curved. Yes, excellent. Like a little boomerang, right? Yes, these have like a little boomerang shape to them. So in this class, then, these guys are going to be able to attack other organisms. So the Vibrio prey on other gram-negatives. Now, it's interesting that they only target gram-negatives. We think that has to do with their cell walls. Because if they try to hit a gram-positive, it has a reinforced cell wall, right? It's bigger. And so it kind of can withstand an impact more. They have to hit another gram-negative because their cell walls are weaker. And that would allow the impact to occur. Yeah.
So is there some kind of chemitaxis that draws them to the gram-negatives?
That's a great question, and it goes right along the lines of how they know to build a sex pelus. It's one of those things where, again, we know they communicate with each other. We're not sure what it is, but you're right. We suspect it's some sort of a chemical release, like a chemotaxis, that they're following a chemical trail of their prey until they can get close enough. Again, like perfect little vampires. Now that gets us to our final group of our gram-negatives, which are the epsilons. And the epsilons are ones that a lot of times are found in microaerophilic environments. So what does that mean again? If I tell you it's a microaerophile, what does it prefer? Yeah, lower oxygen, right? So these are ones that like to be in areas that have a lower oxygen level. Also, a lot of times they prefer higher CO2. So lower oxygen, higher CO2. There's two main organisms in here. There's Campylobacter, which can cause lung infections. So it will cause deep lung infections, deep nair infections. And then Helicobacter, which is one that's associated with causing ulcers. Now, the Helicobacter has a really interesting history to it. For the longest time, we thought that people got ulcers because they were stressed. And that stress led to the creation of these little holes in their stomach lining. That's basically what an ulcer is. An ulcer is where your stomach lining starts to have some sort of a fissure or a tear in it. And now the stomach acid that's in your stomach will be able to touch tissue that's not protected against it. And so you feel this burning sensation or this pain sensation. That's the feeling of the ulcer. So there's a long time that we thought that stress was related to ulcers, and it is in a manner of speaking. I mentioned it here before that when we get stressed, something happens to our immune system. What happens to it? Yeah, it lowers, it gets weaker. So I mentioned that during finals week, during midterms, everybody starts to feel a little run down, right? It's because we're all getting stressed out. Our immune systems are dropping a little bit. So in this case, our Helicobacter pylori is able to sneak in and start to establish itself because of a reduced or lower immune system. Now, how we get exposed in the first place, it can be through food, it can be through drink, it can be through environment. If somebody near us has it, they handle something and we handle it and then put it in our mouths, we can get exposed that way. It's one that's easily spread. But if you have a healthy immune system, you can usually knock it out. The problem with Helicobacter pylori is once it starts to make little holes in your stomach lining, it can be harder to treat. And it can be treated. It's a process. You have to take several capsules that will kill it off. But once you get rid of it, it can come back. And we worry about patients who have reoccurring infections. It seems like some people are kind of prone to it, like their immune system just never really learns to defend themselves. And if you keep getting ulcers, you want to get checked because it can lead to stomach cancers. Having that tissue exposed to acid is just not healthy for it. It's a mutagen, and it increases mutations. Now, there's a really interesting story behind the Helicobacter, and that has to do with Professor Barry Marshall, who is an Australian researcher. He and his lab were the first to put together this link between ulcers and Helicobacter pylori. And the reason he did this is he was studying patient samples, trying to figure out if there was an organism behind the ulcer. And he started noticing this little organism that was popping up. Of course, it was a gram-negative, little vibrio shape. So he decided to show and publish a paper showing his findings. And he did so, and he was laughed at. People were like, bacteria causing ulcers? That's crazy. That's never going to happen. And so he was like kind of ridiculed. Now, for a lot of people, that ridicule would have been embarrassing, but they would have slowly stayed behind the scenes and kept trying, but not Professor Marshall. He was mad. So he went back to his lab and he went to his doctor and he had an upper GI scan to confirm that he had absolutely no signs of ulcers. He said, "Are you sure? I want you to make sure you take all of these pictures, no ulcers." And then what do you think he did? He went back to his lab. There was a flask of Helicobacter pylori that he grew. What do you think he did with that flask? Yeah, he drank it. He picked up a flask and drank 500 mils of straight Helicobacter pylori. Yeah. So a couple days go by, he starts feeling not so great. A few weeks go by, he's feeling some pain. He's got some major stomach pain. He goes to his doctor. He has them do another upper GI. The doctor's like, "Why are you here? We already said you're fine." He's like, No, I want you to check again. So they do another upper GI, and he is full of ulcers to the point that he was actually anemic. He was losing so much blood. I mean, he almost killed himself doing this. And the doctor's like, "You are full of ulcers." And he's like, "That's great." And the doctor's like, "I have no idea what's wrong with you right now." He was like, he's trying to figure this out. But he published. He's like, "Take pictures and I'm going to publish it." And he published his article with his own pictures. And finally, the medical community had to listen to what he was saying. I mean, that's pretty extreme. That's a person who is passionate about science, right? If you can be that passionate about your job, you're going to be fine. because you're never going to work a day in your life when you have that much passion behind your work. The other organism I mentioned that's in the epsilon group is the Campylobacter. And Campylobacter, I said, can cause upper respiration infections. If it gets into our GI tract, it can also cause intestinal infections. Now, the reason we worry about Campylobacter is it can cause something called Guillain-Barré syndrome. Has anybody ever heard of that? Yeah, Aaron. Do you know what?
Neurological disorder.
It is a neurological disorder, yes. And so what it is it's an autoimmune neurological disorder. So our neurons normally, as you guys know, have taken physiology, they have those myelin sheaths around them. For some reason, when we're exposed to certain infections, our bodies will start attacking the bacterium. And if that bacterium is somewhat similar to our own cells, our body gets confused and starts attacking our own cells. Unfortunately, Campylobacter is one that has a trace pattern similar to a myelin sheath. So some people's bodies will attack the Campylobacter and then attack the myelin around their nerves. And so they'll start having issues where they're starting to have tingling sensations. It feels like their legs are always asleep. For example, you know that pins and needles after your leg falls asleep or something? They feel that 24/7. Sometimes it'll go away. So sometimes it can go away within a month. Sometimes it'll go away within a year. Sometimes it never goes away. And the problem is we can't tell. It's all based on a person's own immune system. So we just never know how it's going to work. So it's one of those things that's always kind of frustrating. There was a mention. So Mishala mentioned that she's been hearing about it with the COVID-19 vaccination. So again, there's always risk with any vaccination that some people can have autoimmune issues. I haven't heard of Guillain-Barré with it. I've heard of it with the flu vaccine. I haven't heard of it with COVID. I'll have to check into it. But again, there's no perfect vaccination for everybody out there. There's always this very slight risk of things that can occur. So that gets us through the proteobacteria group. The last little area we want to talk about are the non-proteos. These are still gram-negatives, but they were ones that were discovered later. And so they didn't really, get found early enough to put them in one of the main groupings. Also, they don't really fit with the other groupings. They're not like an alpha, they're not like a beta, they're not like a gamma, they're not like a delta. So we don't really know where to put them. And so they kind of just said, well, they're gram-negatives, we'll just call them the non-proteobacteria. A little lazy, but we just kind of go with it. One of the most important ones in here is chlamydia. Chlamydia is a really common STI. It kind of fights back and forth to be one of our top STIs. It goes back and forth with human papilloma or human warts, genital warts virus. They kind of take turns being the most important. We have an average of about 2.9 million infections each year. Now, one good thing about COVID, our STI infection rates dropped dramatically because people weren't going out, right? I mean, people were staying home. If you stay home, you don't get an STI, apparently. So our STI rates did drop, but we are starting to see increases again. So it never really goes away. It's always around and always visiting us. The interesting thing about this is a lot of these cells are very specific about the cells they can target. So, for example, if I have chlamydia, it has to infect columnar epithelial cells. If I have a hysterectomy, for example, and I remove parts of the inner workings or the parts of the uterus and things like that, I remove a lot of the cells that they can infect, which means that I no longer have to have STI screening for certain organisms. This is why if you've ever had any kind of surgery like that, your doctor might tell you, oh, you don't have to get screened for this anymore. It's because you no longer have the cells that they can infect. Kind of a bonus, a little good thing. Okay, let's talk about our gram-positives. So our gram-positive bacteria can be separated, and I mentioned this before, based on their DNA. So they can either have a high amount of A and T, and if I have lots of A and T, then that means I have low G and C, or I can have a high amount of G and C and low A and T. So basically, I'm looking to see what bases are present in the DNA, and then that gives me kind of a fingerprint pattern. There's the phylum, the Firmicutes, and the Firmicutes are going to be our ones that have a low GC ratio. So the Firmicutes are the low GCs. And this would include ones that we've seen in here. Bacillus, we have Clostridium, Staphylococcus. Those are going to be low GCs as far as the grouping goes. So if I look at my low GCs then, what I see is some of these can produce endospores, Some of them are wall-less. Remember our mycoplasma friends? They're put in this grouping. And some aren't able to produce endospores. Think of things like our staph and our lactose. They're going to be ones that are just going to be able to live in the environment for the moment, and they don't have that little escape capsule to be able to produce an endospore later. So with Clostridium, let's start with this one. Clostridiums, we've talked about them several times in here. They're obligate anaerobes. They produce endospores, which is how they're able to survive in the environment when they're exposed to oxygen. And there are a lot of Clostridium species out there that can cause disease if they get inside of our bodies. So if we look at this, this is a picture of Clostridium perfringens, and it's in the early stages of gas gangrene. You might have heard of a wound going gangrenous. And so what happened here is, unfortunately, through an injury, some of the endospores were able to get deep into the anaerobic tissue. Now, sometimes this injury can be a surgical site, sometimes it can be a puncture or a bite wound, something that pushes into the deep tissue. Then what happens is when that tissue closes, the organism's endospores hatch out and start to grow. And when they start to grow, as we mentioned in metabolism, sometimes some of the side effects of going through Krebs in our metabolism is we create CO2. Remember we mentioned CO2 being released? Well, those CO2 bubbles are now under your skin. And the more and more that they grow, the more CO2 you start to have build up under the skin, and it starts to swell, and it'll actually cause the skin to split. So this is a very mild picture of the tissue damage. I figured it's early in the morning, we don't need to see anything major. You can look stuff up. So what's going to happen then is that we are going to want to treat this as soon as possible. The easiest way to treat it would be to expose it to oxygen. So a lot of times what will happen with a wound like this is they'll do a debridement, they'll go in and scrub it, clean it, and then open it and put you in an oxygenic chamber, which means they're going to be pumping pure oxygen directly into that wound site. As soon as this organism gets exposed to oxygen, it dies. It goes back into the endospore stage. Then we can administer antibiotics at the same time, which will prevent the endospores from hatching out. It builds you time for your immune system to find the endospores and destroy them, and then you're able to clear your infection. So that's one of the things that we try to do with Clostridium. The risk that we run into with Clostridium then is any type of opening, or another factor would be something like low circulation. Diabetics a lot of times can be prone to an injury that they don't even know that they have. Because of the low circulation, they don't feel the injury. Sometimes diabetics have numb feet or numb extremities. And so they might have stepped on something, had a puncture, and didn't even know it. And so that's one of the things that we can see with a lot of our Clostridiums. Here's another picture of our low GC organisms. This is a picture of MRSA that is unfortunately starting to create kind of a open wound or sore on the skin. The Staphylococcus aureus would be able to do the exact same thing. It's basically where what happens with staph and with MRSA is just a resistant strain of staph, is when they get into our skin, they build this wall around themselves, and picture in your head kind of like an igloo. Has everybody seen an igloo before? Okay, so they build this wall, like a little igloo around their cells. And then our immune system tries to come up and get to the cells, and they can't because there's this little igloo in place. And so what happens then is that we start to get this little pustule. First it looks like a spider bite, and then it starts to grow into an open wound or an oozing sore, and then it keeps growing. And when you see something like this is like an iceberg. We're looking at the tip, but it goes deep into the tissue. It would be quite painful to touch. And under that tissue is this igloo full of pus and staph. The only way to really treat this then is to put the patient on antibiotics and to clean out this wound. So we're going to have to get a scalpel. We're going to have to cut that wound out, basically cut the igloo out. We're going to have to cut around the staph. And when we remove that igloo-like structure, it allows our white blood cells to flood into the area. So as you can imagine, it's painful. If we don't get all of the staph out, it will come back. So that's something that we have to watch for. We have to make sure it's completely clean. Yeah.
So I would imagine it's really difficult for our body to build up antibodies to MRSA.
It really is. Part of the reason is we have staph epi on our skin, and so our body doesn't target staph as quickly. Staph aureus is a little different, and MRSA is a little different, but still in our immune system, it sees the genre and it doesn't panic. It takes a little bit for the organisms to start attacking for our immune system to kind of go, wait a minute, you're not the nice one, you're the mean one. And then they kind of go back to it. It's like the evil twin syndrome, right? It's like it looks similar. And it's really hard to build antibodies too, because a lot of times they just can't get to them. Like we have to break that igloo structure down. And we'll talk about that when we talk about body defense lines and how our organisms evade our immune systems. We'll talk more about that igloo-like structure that they can build. Here's a picture of our staph. Again, the little cocci shells. Another thing we have to worry about in our gram-positives is listeria. And we mentioned listeria, how it's found in meats and cheeses that aren't properly pasteurized. That's why this is up here. Listeria, Clostridium will be found in things like hot dogs, deli meat, sprouts, smoked seafood, things that haven't really been heated or sterilized completely. So we always kind of worry about this. The reason we worry about listeria was because of the fact that if you're pregnant, what can it do? Remember that from my other lecture? What's that? Yeah, it can cause birth defects, or it can even completely kill the baby, because it's one of the few bacteria that can sneak past and go into the transplacental layer. Usually our immune system, it will not allow bacteria to pass towards the baby. It's going to be protecting that umbilical cord and that attachment that the baby has. But unfortunately, listeria is one that's able to sneak inside. And so it gets through before our immune system is able to stop it. Our high GC content. So this would include the group called the actinobacters. Now, one of my favorite ones that's in here is the top one, which is Actinomyces. Actinomyces, it can be a pathogen, but it's also found in soils. There are a few different strains, just like Staph aureus versus Staph epi. But the Actinomyces is one of my favorites because you know the smell of rain when you're outside, and that smell of that earthy soil. I know it's been so long since we've had rain, but you can kind of recall it, right? That really neat earthy smell. That's released from soil-dwelling organisms. It's actually an enzyme that they create called geosin. So it's an enzyme they create called geosinase, but it's on the protein geosin. And what it does with this enzyme is when it gets wet, it vaporizes, and that smell that we get, that earthy smell, the rain smell, is actually due to organisms in the Actinomyces group. So whenever you walk outside and you smell that earthy smell, you can think the Actinomyces. That's just organisms that are living in the soil, which is pretty cool. We do have a few pathogens in this group. We have our mycobacteriums, our nocardias, our streps. So these are ones that can cause illness in humans. And what we can see is that a lot of these can be pretty serious or harder to treat infections as far as when they are able to form deeper in the body. Like Mycobacterium is one that's able to have that fatty acid layer around its cell wall. So because of that, it's harder to treat. Same with nocardias. Streptomyces, they're ones that we can treat pretty well. Actinomyces pretty good, but these are ones that are usually found in the soils. So they tend to not be really pathogenic. Over here, with my ones that are also going to be in the pathogen group, I have this guy, Propionibacterium, and that's going to be the bane of a lot of teenagers' existence because Propionibacterium is associated with acne. If you ever fought with acne when you were a teen, you can thank this organism. It is one that is able to grow on our skin, and it actually survives by eating the oil that our skin produces. It eats the sebaceous oil that our skin excretes. Now, what can happen then is as this bacterium starts to grow, it's eating the oil from the pores, and we try to get rid of it by washing our faces more, right? And that's actually the opposite of what we should do, because when we wash our faces more, it dries our skin out, and guess what our skin produces more of? Produces more oil, and that's going to be more food for this bacteria. So usually what we need to do is we need to kind of treat acne from the inside. So what they'll have you do is a lot of times they'll have you go on birth control, or they'll have you take some sort of a supplement or pill. And what that pill or birth control is doing is it's altering the pH of your skin. It changes the pH just a little bit. It makes your pH a little bit more basic. And these organisms, they don't like that pH shift. And so what happens is it's just enough of an alteration that it kills these guys off. So that's how a lot of the acne treatments work. They're neutralizing the pH. That's what the special washes are doing too. They're neutralizing your pH. Now we could go the other way. There's also acid washes. You might have seen those in the store where they say that they're acidic. Again, we're just changing the pH. So we can go either way. Yeah.
Is that bacteria contagious?
It is not contagious. So usually it has to be kind of a genetic predisposition, meaning that we genetically have to make more oil or enough oil to support it. This is why sometimes acne will run in families. This is also why you'll have people that never have had acne. It's because they just don't produce enough oil to be able to support the organism. So some people are naturally just blessed that way. You know, it's just like the luck of the draw. But if it's something that you know runs in your family, you can kind of keep an eye on it. And again, it's something that's easy to switch now that we understand the basis of it. It's much easier to switch to a hormone supplement or some sort of a pill to affect the pH of the skin. So now that we understand what causes it. Used to, we thought it was poor hygiene. We would have people washing their faces and we made it way worse. And now we've learned that actually is like the exact opposite of what to do. Corynebacterium. This is an organism that we actually have vaccinations for, which is known by C. diphtheria. And diphtheria is unfortunately a really nasty organism that starts as a throat infection. And so it'll start with kind of feeling like you have a sore throat. And then over time, that sore throat will get more and more sore. And then all of a sudden, you'll start feeling like you can't breathe. You'll start having a hard time swallowing. What's happening with the C. diphtheria is when it starts to grow, it produces a cotton ball-like effect in the back of the mouth. So if you look here, here's your uvula hanging down. This is the bacterium growing, and it grows in this thick mucus structure, and it's basically like having a chunk of cotton stuck in your throat. And if it's left untreated, this cotton will spread, and it'll move from one side to the other, and it'll actually cut off your respiration. It will grow over your trachea. So you're not going to be able to breathe. And so it can actually result in death. This is why it's one of the vaccinations that we encourage out there. It's one that we are fortunate here because of our high vaccination and our good medical care. We don't see diphtheria too often here. But if you travel to other countries, India, if you travel to Europe, they will have diphtheria circulating. So again, you always want to go to the CDC website if you're going to travel out of the States and see what's prevalent in other countries and what you can do as a preparation before you go. That gets us out of our bacteria, and it gets us to our final domain, which are the archaea. Now, as I mentioned before, the archaea do not cause disease. So I don't really have a whole lot of organisms to introduce here, but I thought we'd talk about a couple that impact us in a positive way. So again, our archaea are going to be the ones that occur in extreme environments. We talked about the hyperthermophiles that can live in boiling hot geysers. We talked about our extreme halophiles. Remember, those are the salt lovers that gave flamingos their pink coloration. And then there's also this middle group, the methanogens. And these are archaea that actually produce natural methane in the earth. They take carbon and they build it to make a methane particle. So if we were to look at our organisms then, we can see some examples of some of them. Our hyperthermophiles, we have one called pyrodictyum. Pyro means fire, so it's the fire dictator. That's kind of cool, pyrodictyum. Our methanogens, we have one named Methanobacter, produces methane. And our extreme halophiles, we have a genus called Halobacterium. Don't you love their names? They make sense, right? The archaea group usually tend to have names that make sense. One of the ones that we are really interested in studying is one called Deinococcus, which to me, doesn't that sound like a dinosaur? You know, like a Deinococcus, it sounds like this cool dinosaur out there. And the reason we're interested in it is it's an organism that is very resistant to any kind of radiation, UV light. It is actually more resistant than even endospores, which is shocking to us. When we try to heat it, it doesn't die. Thankfully, it's not a pathogen, so it's harmless. But we're studying it because we're fascinated as to how the archaea have evolved these amazing strategies to survive. And thankfully, so far, they haven't shared their secrets with bacteria, so that's good. They don't really talk to the bacteria too much, so we're good. Bacteria stay in one field, archaea stay in the other. One of the organisms that's a hyperthermophile is one called Thermus aquatus. And this was found in the boiling hot geysers of Yellowstone. Now, the reason that this is important to us is it's the source of a special enzyme called Taq polymerase. This is what we use when you do PCR. Have you ever heard of PCR? You have polymerizing chain reaction. It's where you take a piece of DNA and you make a whole bunch of copies of it. This is how we amplify DNA for like fingerprints, or if we amplify DNA for like semen left behind at a crime scene. This is the way we can amplify DNA to study it. We have to melt the DNA to make copies of it, and we were worried about trying to find an enzyme that would work to read the DNA that could withstand high heat. And we lucked out because this organism in Yellowstone was able to be adapted to a PCR process. And so now it's grown in labs to be able to harvest the special enzyme from it. So again, it's not anything that harms us, it helps us. The last slide I want to leave you with is one about microbial diversity. And this is just kind of a reminder that we are surrounded by bacteria, and for the most part, they're pretty harmless. If I was to go outside and just take a little scoop of soil, just like a little gram of soil, in that little gram of soil, I'd have about 10,000 different genera of bacteria. That's amazing. So many organisms living in just that little gram of soil. So the reason I bring that up is because that means we have a lot of bacteria out there that are still unknown, that have yet to be cultured, that have yet to be named. So these are ones that we're still not sure what mysteries they hold. And the really cool thing is if you do go further in microbiology and you decide to study organisms, you might be somebody who discovers a brand new one. And you can name them. That'd be pretty cool. Like, I mean, you can name them after your favorite instructor, like, Hadley Eye. I mean, just throwing that out there. I'm just saying, if any of you ever find anything, how cool would that be? I think we need a Hadley Eye, right? How cool would that be? Anyway, so just a reminder that bacteria and archaea are all around us, and for the most part, they leave us alone. There's only those few pathogens that we kind of briefly glimpsed into that really cause us so much grief in our life. Okay, so I'm going to stop my share here, and we're going to jump to our next category, which is going to be our eukaryotes. Start talking about these guys. And your virus slides are now available as well, so you can go in and check out your virus slides. They're up now. So the eukaryotes are going to be organisms that are kind of like a lot of what we've seen with our bacteria. Some can cause disease, some are harmless, so we're going to talk about different groupings with them. But these are going to be ones that are much harder to treat. And the reason they're harder to treat is they're similar to our own cells. So what do eukaryotes have that prokaryotes don't? What's that? They have something in the center of them that prokaryotes don't. The nucleus. Thank you. I almost had a heart attack. I was like, no, you guys, don't forget. Yeah, they have a nucleus. Excellent. Whew, okay. Yeah, they have the nucleus. And so, let me make sure I've got my chat up here. Okay. And so, we're very curious to see how we can treat these organisms without hurting our own cells. Because their cells are so similar to ours, we no longer have that magic bullet concept. I can't target them as easily because if I'm hurting one of their enzymes, a lot of times I have the same enzyme. So this is where I start to run into problems with treatment. So as far as how they're classified, well, they're separated into a few main categories. So we're going to have things that are going to be distinguishing characteristics of organisms like algae, protozoa, fungi, and we'll talk about each of these as we work our way through. We'll talk about how these things are able to attack us, and we'll talk about some of the most common pathogens that are out there. Now, this is kind of a shocking estimate, but it's estimated by the CDC that at any given time, about half of our population on the planet has some sort of a eukaryotic pathogen in or on them. That's huge. That's like a scary number. But then we kind of start to think about what that means. And what that means is that can include things like toenail fungus. That can include things like athlete's foot. That can include things like a yeast infection. Things that are very, very common. So it starts to make a little bit more sense. It's like, okay, yeah, maybe I can buy into that. Because those are infections that we constantly get exposed to or fight with, and they get cleared up and they go away. So there's this idea then that eukaryotes are kind of all around us, and they can cause all sorts of issues if they get inside of us. Now, some of the organisms that are out there are very different looking from us. Things like yeast look very, very different. But other organisms are going to be somewhat more animal-like our protozoans. They tend to be almost like little animal cells. So some are going to look similar to what our cells would look like under a microscope. So we're going to start with our fungal group, our fungi. And if you are somebody who studies fungi, then you would be referred to as a mycologist. A mycologist is the study of mycology, which is the study of fungi. We have a lot of different types of fungi out there that can impact us for better or for worse. All of these organisms are chemoheterotrophs, meaning that they get their nutrients from other cells, just like our pathogens, just like us. A lot of them are what we call saprophytes. And what that means is that they're going to live on another organism. So if you think of something like athlete's foot, it's not living inside of us, it's living on us. So that would be an example of a saprophyte, something that lives on top of us. Most of these organisms are either aerobes or facultative anaerobes. As I mentioned, there's a few pathogens. Some can live unicellular as our yeast. So yeast are in the fungi kingdom. And then we can also have some that are more filamentous, thread-like, and that would include our molds. So if we start to look at these organisms, we can just see at the bottom here that they have very different appearances based on what type of organism or what category they're in. So just because they're in the kingdom fungi doesn't mean that they all look the same. They'll have variations depending on which genus or species that we're looking at. So for example, we can have our yeast, which are usually candidas, which are these little circular, single cells, or also have like a little cell attached to them. I mentioned that they'll do budding, where they create their daughter cell and don't separate. And then we can also have some that are molds that are going to grow like little filaments out there. So a lot of them will have different appearances. Now, as far as how most of our molds and fungi are going to reproduce, most of them will be reproduced through processes of growing hyphae, which are little thread-like extensions. And in these hyphae, they'll be able to create what are called kinidiospores. And the kinidiospores will be able to help them with reproduction. Now, hyphae come in two different types. We can have septate hyphae. And what this means is it's like a hyphae or a thread extension with walls in between it. Think of it like a house with little individual rooms. We have walls in between. And I'll show you a picture of that in just a second. Or we can have coenocytic hyphae, and these are hyphae that instead of having cross walls are just fused as one long thread. So let's look at a picture and see what that means. So there's my septated hyphae. Do you see how I have little cross walls? So like individual rooms separating them. And so with my septated hyphae, a lot of times what can happen is if one of the pieces breaks off, it can actually start a whole new mold, or it can start a whole new fungus, because it's like its own little room, right? And if I break it away, it doesn't matter. It can separate easily. We call that fragmentation, so it can fragment off. If I'm attached, if I'm coenocytic, that means I'm sharing my nutrients, and then if I break it off a piece, it can actually negatively impact the mold. So the molds sometimes can either break apart to reproduce, or they can't, and they can only use spores. So they have two different varieties that they can do. The other thing that they can produce is they can produce either aerial hyphae or vegetative hyphae. Aerial hyphae, these are again thread-like extensions, hyphae or threads, are ones that can grow straight up in the air. Do you see that aerial hyphae at the top there? These sometimes will look like an old man's beard. They'll shoot up. And we have to be careful when we're growing these in the lab. A lot of times when we grow different types of fungus, we actually have to tape the lids of the petri dishes down. Because otherwise you'll come in the next day and they've grown so tall they've pushed their lids off. And so they'll have lids like laying everywhere. So we have to be careful with growing them. We have to like tape their lids in place. So aerial hyphae will grow up. Aerial hyphae tend to be the ones that are going to be used for reproduction. If I grow up, I'll have little spores on the end, and those little spores can now catch the wind currents or the air currents and they can spread. Vegetative hyphae are ones that grow straight out. Their job is to find food. So they're going to grow out and try to tap into nutrients. They're going to stay flat against the auger as I'm growing it and take and absorb all the nutrients that are present. So aerials are reproduction. When I see vegetative, those are going to be ones for food. So here's a picture of how a hyphae can start. I can have a little spore that'll land in an area that it likes. It'll start to grow its little hyphae. Is this a septate or a coenocytic hyphae? Coenocytic, good, because it's all fused together. So this little hyphae is going to grow. Is it aerial or vegetative? Aerial, excellent, because it's growing straight up. If I was vegetative, I'd be growing straight out. Okay, so I'm aerial, so I'm growing straight up. And so this is going to be a hyphae that eventually will be used for reproduction. Okay, so we can tell a lot to say how it grows. Now, once those hyphae start to accumulate, so I have a whole bunch of threads together, I can now refer to that hyphae as a mycelium. A mycelium just basically means a whole bunch of hyphae together. Okay, so these mycelium then, as I mentioned, can either be flat against the surface, that's again vegetative hyphae. If it's a whole bunch of them, we can say vegetative mycelium. And their job is to collect nutrients. If I grow straight up in the air, then I can be an aerial hyphae or an aerial mycelium. And what's going to happen here is I'm going to produce spores for reproduction. So just different ways that my molds can grow. And we've seen this. For example, you've seen bread mold. And if you let bread mold sit, it will get fuzzier and fuzzier growing upwards. It's producing more spores growing upwards to reproduce, but it's also spreading out on the bread. So you see vegetative as well as aerial. Okay, so you see both factors. If you don't believe me, let some bread mold. You'll get to see it. Now, in this same group of our fungi, we also have our yeast. And when we look at our yeast, they look very different. You might be wondering, well, why are they in here? They're not really filamentous. If anything, they tend to be either single-celled or they have a daughter attached to them. They tend to produce hyphae that are often referred to as pseudohyphae, meaning that they're not really true hyphae. They're sharing nutrients with their daughters there. They tend to be facultative anaerobes. They can grow with or without oxygen. So you might be looking at these going, well, how is this related to my bread mold? Well, the reason it's related to bread mold has to do with the type of cell wall it has. It has a similar cell wall. And then also it has to do with the temperature that the yeast is grown at. It turns out that if I grow yeast at a high temperature, it will tend to have kind of that single circular appearance, very orderly, and almost look like a bacterium. It looks very similar. I mean, that last picture that we saw was kind of like a bacterium look to it, right? If I go back one, it almost looks bacterial, right? But it turns out that if I grow yeast at a colder temperature, like room temp or below, it tends to elongate and flatten, and it looks almost like a filament. So when we first started studying yeast, we were studying it at room temperature, and it looked like a mold. So that's how they also got kind of grouped into this category. So what happens with them then is, remember they go through mitosis to reproduce, and then I told you they often don't go through cytokinesis, so the daughter cell stays attached to them. And we say that if that bud forms, if the daughter forms and doesn't leave, it's forming a pseudohyphae. It's forming a pseudohyphae. And so when that happens, what we're going to see then is that the bud is going to fail to separate. And so when it fails to separate, the mother is going to kind of start to support the daughter. This is a short-term support though, because what will happen is even though that daughter stays stuck, eventually the cells that are present are going to close up their cytoplasm and start to function independently of each other. So even though I have my daughter attached, I'm no longer supporting her. It's kind of like how our parents cut us off. You know, at a certain age, where they're like, okay, you've had enough, like you're done. And they stop supporting us. So rude, you know. Okay, so they stop supporting us. And so when that happens, we're still attached to them. You're always still attached to your parent, right? You never like forget them. You don't leave and forget them. But they're no longer supporting you. And so that's what happens here. That's what we call it a pseudohyphae. They're still attached. Originally they were sharing nutrients, but then we build a little wall between them and they stop with the sharing. Here's a picture of yeast growing at different environmental temperatures. So if you see yeast growing at warmer temperatures, they look kind of like bacteria. Here's yeast grown though at room temp. Do you see how it's more filamentous? It's long and skinny. So it looks more like a true mold. So it's just long and skinny when we look at it. The life cycle of fungi. So fungi can reproduce, as we mentioned, through kinetiospores. That's one way that they can reproduce. And if you look at a kinetiospore, it looks almost like a lollipop structure. So this is the little aerial hyphae. That's the little thread that's coming up. And then all of these little dots here, those are all of our kinetiospores. So for my group that's at home, this is the little aerial hyphae, and then these would be all of the little kinetiospores that are present. And so what's going to happen then is with these little spores, if there's any kind of wind or motion around, those little spores are kind of like dandelion seeds. What happens when you pick a dried out dandelion and blow it? Please tell me you've all done that as a kid, right? It goes poof and it like disappears, right? If you haven't done that, walk around campus and find a dried dandelion. It's time to do it, all right? And so just like a dandelion, those seeds go poof and they go free in the environment and they spread dandelions all over your yard, right? This is why when you were a kid, your parents would tell you, don't do that because you're spreading dandelions everywhere, right? Same idea is true here with our molds. When they get near an air current, they go poof and they just spread all around. This is why no matter how clean you are, you will always find mold in your kitchen. Somewhere, on the surfaces, somewhere. We can always find it. And it might not be actively growing, but the spores are present. Okay, so it's like it comes in our hair, it comes in on our clothes. It's just always there. It comes in on our groceries. No matter how careful we are, we always are going to have spores around us in the kitchen and in the area. Now, as far as the spores go, when they're released, they're like exact little replicas of their parent cell. They're little clones. And so they're going to create exact copies of the cell that released them. So they're going to just poof into the environment and then create an exact copy. Another thing that we can see is that there's different kinds of fungi out there. And so we started to kind of categorize, as biologists do, our different types of fungi into different groupings. So our first grouping were the main group called Zygomycota. When you hear Zygomycota, think of bread molds. So these are going to be, for example, ones that we find in our homes a lot. So when we look around our home, or if I take a spore sample from a kitchen counter, a lot of times I will find the presence of Zygomycotas. The most common one is the official bread mold, which is Rhizopus, and that's that greenish kind of stuff that grows on our bread, that greenish white that grows. The next grouping we have are the Ascomycotas, and these are sometimes referred to as the sac fungi. And the reason for that is when they do their reproductive organs start to make their little spores, they have a little sac that's kind of around the spore to help keep the spore from drying out. So when I look at my sac fungi, they have this extra little structure around their spores. We also put yeast in here because yeast, when it was first being studied, was circular and they assumed it was in a sac. They found out later it's not really a sac, but they kept it in this family because it really didn't fit anywhere else. So when you hear Ascomycotas, think of a sac. Different ways to remember this. You can think of you're asking for a piece of or some yeast, so Ascomycota, just different ways to try to like keep track of who's who. The Cydiomycota, these are the ones that are referred to as the club fungi. And they look like little clubs that you would hit people over the head with in medieval times. Don't you love the violence? It's like, oh. So they look like they have like kind of like almost like a mace-like appearance. They come out and they have these little balls with spikes at the end. These are all their little spores. And these are going to be ones that are going to grow in the environment, and they create kind of like almost like a pedestal or a stalk. And then they kind of release spores into the environment that way. An example of these would be mushrooms. You ever seen mushrooms, how they grow like a little umbrella? If I was to look at the umbrella, and if I took a gill out, the little slits that are under the umbrella, this is what I would see. I would see that they have these little club-like extensions growing underneath that little umbrella, which is pretty cool. A lot of people think that mushrooms are plants. They are not. They are actually a type of fungus. So here's a picture then of our three groupings. Our Zygomycotas, which are our bread molds. These are also ones that we would see in our house spoiling our veggies. These are the ones that are most commonly in our home. We have our Ascomycotas. Again, these are going to be like our sac-like fungi. this is not a typical mushroom that's here. It's a special type of mushroom, a truffle, so it's a little bit different. It's not in the typical mushroom family. And then we have our Basidiomycotas, which are our true mushrooms and our true club fungus. So let's talk about fungi in regards to pathogens. Well, there's a lot of different ones out there. So the first one we're going to talk about are the ones that affect plants, and these in turn can affect us. So one of the ones is rust disease. If you've ever grown roses around here, a lot of times on the back of your rose leaves, you'll see bright orange spots. You guys ever seen that on a rose bush? You see these bright orange spots? They're like fluorescent orange. And it's referred to as an infection called rust. And what that is it's a fungus that's growing on our rose plants. It tends to be common here because we get a lot of fog as a moisture, and any moisture that sits on the leaves for a long term will allow this fungus to start to grow. The easiest way to treat it then is to expose the rose bush to an antifungal. And I bring that one up because a lot of us have seen it. You might have seen a rose bush that has these bright orange spots and been like, what is this? It's a type of rust. The next one is Dutch elm disease. Dutch elm disease, and you can see a town here that had a whole bunch of elm trees, and then at the top you see all the elm trees, and then this is it just after a few months as the trees started to die off. So this is like within the span of about 9 months. They were down to just a few trees. Dutch elm disease was a disease that wiped out about 40 million trees in the early 70s. It was brought in by accident, and it was a fungal infection that was brought in on elm wood that we pulled in from another country. And unfortunately, these spores were released and they got into our wild Dutch elm population. Now, what this does is it causes a vascular disease in the tree, which means that their bark starts to peel off. And so as you can imagine, that would be like us losing our skin. And so then they're exposed to everything out there. So it can cause mass death. Now, unfortunately for us, Dutch elms are actually really social trees. And I know that sounds weird, but when a Dutch elm starts to grow, its roots will spread out, and those roots will actually entwine with its neighboring trees, with the neighboring Dutch elms that are nearby. And when it drops seed pods and when their baby trees grow, the mother tree's roots will actually wrap around the baby tree's roots, and it will share nutrients. So these are like very nurturing trees. They have a whole ecosystem underground. The problem with that then was when one of these trees got infected, it would wipe out all the trees attached to it, because unfortunately it would go through the roots, through the vascular system, and spread to everybody that was nearby. So if a mother tree was infected, all of her babies were infected, and whatever ones they had tapped into were infected as well. So it was a really devastating disease when it occurred. One of the ways we got rid of it was through grafting. We found that there was a natural Dutch elm in Europe that was naturally resistant. And so they actually brought in parts of that tree and grafted it onto the Dutch elm that were exposed, and they got immunity that way. That's pretty cool, right? It's kind of like an early grafting from the 70s. That's pretty cool. Other impacts, ergot poisoning. Has anybody ever heard of ergot? Aaron has, yeah. Ergot poisoning is something that affected us in the past. Have you guys ever heard of the Salem witch trials? Yeah, ergot poisoning we suspect was the cause of the Salem witch trials. We'll watch a video about it in here. It's pretty cool, but basically the synopsis is ergot is a fungus that grows on rye or wheat. And if you ingest it, it is actually the precursor of LSD. It puts you on an acid trip. Can you imagine the Puritans on an acid trip? I mean, your mind just boggles at that, right? So that's essentially what we think happened with the Salem witch trials. We think that the conditions that year, they had a really wet summer and then a really dry spell and then followed by another wet summer. It was like perfect growing conditions for this organism. And they wouldn't have seen it when they were harvesting. I mean, this is the wheat, and this is what ergot looks like. And if you have a whole bunch of it, you wouldn't even notice it. And if you harvest and grind it up and make bread and then eat it, you're consuming more and more levels of LSD every time you eat your bread. Some of the signs and symptoms are chills. You start to feel like somebody's watching you. You get like paranoia. You see swirling figures. You see things like, it's like an acid trip. You see things like coming out of the ceiling at you. They are perfect descriptions of what the Salem witch trial people were saying that they were going through. Later on, we know it became a popularity contest. That was eventually what happened with the Salem witch trials. They kept progressing. They had people who were killed or tried as witches until one of the governor's daughters was accused of witchcraft. And then all of a sudden the fun and games stopped. The governor was like, that's it, we've had enough. We're not doing this anymore. And he's like, this is all hearsay, this is all crazy, and that's when everything kind of stopped. And so we think, though, that what started it was LSD exposure through ergot. It was found out by accident. There was a researcher who was growing ergot in his lab, and he started, throwing away some of the old cultures. And he usually would wear gloves when he grew and stuff. He didn't have his gloves on that day. He was in a hurry, and the petri dish slipped, and he ended up getting some of it directly on his hand. And he was like an old hippie guy. He'd been through LSD, and he was like, all of a sudden he had this acid trip, and he was like, what is going on? So what did he do? He went and did it again the next day to see if it was really bad. Yeah, Brianna.
So it's not what's in LSD, but it's like
It has a component that is similar to LSD. So it's not LSD itself, but it is a component that is similar. It has the same neurological effects, where it makes our brain just go, woo! It kind of goes off on a crazy trip. Another fungus that affected us was the one that caused the Irish potato famine. So when this occurred, people had planted their potato crops. That was their main food source. Their potatoes started to grow. And unfortunately, this type of fungus, when it gets into a potato, instead of growing a potato that's like 5 or 6 inches in size, it creates a potato that's maybe about an inch, really, really tiny. And it doesn't have any of the starch storage inside of it. You can't tell by looking at the plant itself. So this is what caused the Great Potato Famine, was people went and harvested their crop, and when they dug up their plants ready to have food for the winter, their plants had no food. They had these little teeny tiny potatoes. And that led to a huge immigration, a leaving of Ireland and coming to the US. And that's where we saw a lot of our population start to increase here, was because we had this leaving because people didn't have enough food. Now, in the same group as ergot poisoning is this really interesting organism called cordyceps. And this is like something out of a horror movie. And so cordyceps is this pathogen that infects insects. It's a fungal pathogen that infects insects. Thankfully, we have never found a human infection, which is good. Now, what happens is this fungal pathogen is able to land on the insect as a little spore, and it will actually get into the insects through its little exoskeleton and into the insect's internal insides. And it will slowly move to the brain of the insect, and it will take over the insect's brain. It's like Invasion of the Body Snatchers. Now, the worst thing about this is the insect seems to be aware of the fact that it now has something in its brain. It seems to start to show signs of stress, but it can't control what it's doing. So imagine watching somebody use your body to do stuff and you have no control. Like, that has to be horrific, right? So what cordyceps does then is it will make this insect leave its hive. So for example, an ant, it'll leave its hive, and it will crawl right above the colony, and it will take its mandibles and bite onto a branch. And now the ant will slowly stay there and starve to death. And the cordyceps will continue to grow inside of it. It'll keep growing in its brain until it pops out through its head as a hyphae extension. It will use the ant as a food source, and the ant slowly starves. And then what happens is the hyphae pops all these chlamydospores out, and it has carefully made the ant attach itself right above the hive, or the home of the ant. And so now all of these spores get released and fall down on the ant's relatives. And then it repeats the cycle. Every ant that gets infected, it's going to do the same thing to it. Isn't that just something awful? It's like, seriously, out of a horror movie. So here's a picture of a grasshopper that was infected. So you can see all of the cordyceps hyphae start to grow. I mean, this is something that's like, like Hollywood could totally take this and turn this into a really crazy story. There's another picture of an ant with a hyphae growing out of it. So it's a really nasty insect pathogen. It can infect ants, bees, it can infect crickets, grasshoppers, anything, any kind of a social insect, it can infect them. Okay, let's talk a little bit more about other types of fungi. So
I mentioned that one of the ways we can kind of separate fungi, the ascomycota, the zygomycota, the basidiomycota, was based on what their spore structure looked like, whether it was a club shape or in a sac. We also have another group, though, that are considered to be asexual. And this means that they never reproduce. They never produce spores. And so an example of this would be something like penicillium. Penicillium, which we know makes penicillin, is what's referred to as an anamorph. And what that means is it's the same organism over and over again that's lost its ability to reproduce. It will make an exact copy of itself. So in other words, penicillin survives by cloning itself. It never does any kind of sexual variation. This is why it was a really good drug for us to use, because we never saw mutations or variations. This is why the drug always worked for us, because the organism that gave us penicillium never changed. This is also why bacteria were able to build resistance to it, because it never changed. So it's one of those ones that just kind of keeps growing, making little clones of itself. It's a very odd thing. Usually nature likes to have mutations. We just don't see that with penicillium. It just doesn't really seem to do that. Now, with our fungi, some of them really prefer to have environments with low pH. So a lot of times if we do have a fungal infection, it'll be found in regions that have different pHs than other parts of our body. So for example, fungal infections on the feet. A lot of times our feet tend to be a little more acidic than the rest of our body just because of sweat and things like that. The groin area, again, can have sweat, so it tends to be a little more acidic. Sometimes the scalp can get too acidic. We talked about how you can get dandruff and you can use Head and Shoulders. Head and Shoulders affects the pH. It makes it a little more basic and the yeast goes, oh, I don't like this, and it leaves. It also releases those metal compounds that make it harder for the yeast to reproduce. So that's how we can get rid of it. They can also grow in areas that have low moisture. So if you think about them on your feet, like if you have athlete's foot, they grow really quickly when it's moist, when you're wearing your socks, but when you take your socks off and they dry out, they're still there. It doesn't kill them. So they can do a lot of different types of osmotic pressure. Some of our fungi that are out there have actually formed some pretty cool relationships with other organisms. If you ever go hiking, you might see lichens on rocks. Lichens are kind of like crusty looking, like little, almost like a plant-like structure. I'll show you a picture on the next slide. And what a lichen is it's an example of what we call a mutualistic relationship. Mutualism in biology means that both organisms are benefiting, or a plus-plus relationship. So in this case, we have our green algae. We have a type of algae that is able to do photosynthesis. So it's producing food from sunlight. We have our fungus, who we said was a heterotroph, who can't make its own food, but it joins this relationship with this algae. And what it does is the algae makes the food for both of them, and in return, the fungus makes a house-like structure for the algae to be protected in. The fungus, as I mentioned, is able to go from dry to moist. Algae needed to be moist all the time, so this fungus will trap moisture and give it to the algae. It will take care of the algae, and in return, the algae gives it food. So everybody has a little beneficial relationship here. It's this little plus-plus thing. If I was to try to separate them, neither of them would survive alone. They have evolved to where they have to have each other, kind of like how we need our normal flora. So if we look at our lichen then, this is what the lichen looks like. Like I said, if you go hiking, you see them on rocks and stuff. If you look at them, what you can see is that they'll have an algae layer. So that's the one that's using the sunlight to make food. And then they'll have the fungus wrapped around them, which is like their little home. So it's kind of neat that they grow together. Yeah, what is that called again when they have like a mutual relationship? Mutualism. Yeah, so you're right on point there. So they're mutualism. So it's pretty cute to see these little guys together. Now, the cool thing about fungi with their association with the lichens is that we can actually use them to tell us air quality. At the Los Angeles Museum, up on the roof of it, they have a whole bunch of a whole bunch of lichen that they've collected, and they can tell the smog levels and the safety levels based on the color of the lichen daily. So when the lichen is starting to get too much of acidic conditions, it'll turn kind of a bright reddish yellow. And then when the acid in the smog starts to go away, it'll start to relax to like a green color. So they can actually use their color indicators to tell air quality. That's kind of cute, right? Makes you want to have a pet lichen. Like people used to have pet rocks. It's like, I want a pet lichen. You know, what kind of day is it going to be? It's like, tell me the day. You know, that's kind of cool. So it gives you like a little pH indicator. In general, fungi are also really good because a lot of them that grow in our environment are going to help us recycle nutrients. They're going to help with plant growth. They're going to help give plants nutrients. We use them in things like food and beverages. We're going to do a lab in here called the Discovery Box Lab, which is going to talk about all the different ways that microbes help us by breaking down a lot of different components in our lives. Some of the things are always kind of shocking to realize, like, oh wow, I didn't even know there were microbes in here. So we'll be doing that lab pretty soon. They help us with vaccine production, drug production, and then they also help us with pest control. So we can harvest things like that, organisms that are attacking the insects, and we can use them to try to kill insects that way. So by exposing them to spores like the cordyceps. Of course, with everything that's good, there's always the chance that there's something bad. So, for example, they are known to cause food spoilage. So that's always an issue. They can cause crop death. So some of them are good for plants. Unfortunately, some will attack plants. They can cause human issues. We'll talk about our human pathogens in a minute. They can cause nosocomial infections. What does that mean again, that term nosocomial?
From the hospital.
From medical or from hospital. Excellent. Yeah, they can cause nosocomial infections. So I go in with one thing, and while I'm in the hospital, I get exposed to something else. So sometimes we'll have patients that go in, maybe they use a dirty shower and they come out with half-baked foot. Maybe they are on antibiotics. We wipe out their normal flora bacteria, and then they have yeast in their body that start to overgrow. So now they end up with thrush, or they end up with a yeast infection. So we always have to make sure that we're monitoring the patients closely, that we're not causing them to have something else that can occur. One of our biggest concerns is immunocompromised patients getting exposed. As I mentioned, these are cells that are very similar to our own. And so if somebody's immune system is already having a hard time and we're limited with treatment, it can be very hard to get rid of fungal infections. Remember, we talked about Candida auris, that emerging pathogen. A lot of you are nodding. That was resistant to a myriad of drugs. We're still watching that one closely, Candida auris. I think I told you there was a recent outbreak, it was last year in Los Angeles at UCLA. So we're still watching it. We're still trying to make sure that it doesn't spread much farther. Now, as I mentioned, unfortunately, some of these also are human pathogens. So we're going to visit some of our human pathogens. When we have a fungal infection, one of the terms that we'll use is we'll say that we have a mycosis. So a mycosis means that we have some sort of fungal infection that we're fighting. These infections can sometimes be in our blood or in our organs. So think of like a yeast infection, it's internal, think of something inside. Or they can be superficial, where they could be like a yeast rash, or they could be like athlete's foot. So something that's on the outside of the body that we can readily see, or something like a ringworm. One of the things that's hard with these is a lot of the fungus that's out there tends to be slow growing. So it's hard to even notice you have an infection until it's already established. And then a lot of the treatments are slow processing because we don't want to hurt your cells while we're killing off the fungus. If we were to blast the fungus with a huge dose of an antibiotic, we would hurt you too. So we have to do it a little at a time. This is why if you've ever had like toenail fungus, it takes forever to get rid of it. You have to like do this special nutrient, you have to paint the nails, and you have to keep painting them. It's like months before it finally clears up because it takes forever for that treatment. If they gave you something stronger, it would actually affect your own fingers. So it's like a balancing. So it can be difficult. So if we see fungus growing on the skin, that's referred to as a cutaneous mycosis. That means I have a skin fungal infection. If I see something that's affecting like the hair or the nails, that means the organism is able to produce the enzyme keratinase. And what that means is it's breaking down the keratin that's in our hair and our nails. If I see something that is like ringworm, that's usually going to be caused by an organism referred to as Trichophyton rubrum. Trichophyton rubrum, Trichophyton is the genus, rubrum is the species, and it is associated with ringworm athlete's foot, and jock itch. It has the name tinea capitis. What that means is fungal infection on the scalp. Okay, so a lot of times if I ask an exam question, I could ask which organism causes ringworm. I'm looking for Trichophyte rubrum because that's the genus and species. But a lot of times students will tell me tinea capitis. Tinea capitis isn't the organism. That's telling me where the fungal infection is. So tinea capitis means infection on the cap or on the scalp. All right. Tinea pedis means fungal infection on the foot. So this is telling me where the infection is, but that's not the genus and species. Okay, so be very clear with that. Candida albicans. We mentioned that we can use Candida albicans in food. It's actually an organism that we use in breads. But it also, if it starts to grow at a high dose in the body, it can result in things like thrush. We see this a lot of times in infants. They'll get like a white-coated tongue, thrush. Sometimes adults will get thrush as well. We usually see that in immunocompromised people. It can make your tongue feel raw and very painful to swallow. It can be treated, but again, it just takes a little bit of time to get rid of the fungal infection. We can also end up with vaginal infections with yeast. A lot of times with women, we have to be careful when we take antibiotics. If you take an antibiotic, it's going to kill good bacteria with bad. And when that happens, that can affect the normal flora in our body. We take away the bacteria. And our bacteria and our yeast, because all of us have a little bit of yeast, are usually in balance. If I take one away, though, now the yeast doesn't have anybody to keep it in balance, it's going to overgrow. And that's why we tend to see people that are all of a sudden getting yeast infections when they're on antibiotics. We have to treat them to try to make sure that doesn't happen. And again, treatments can be challenging. This is an example of tinea capitis, or a ringworm on the head. So if you try to treat the patient, first of all, notice they've had hair loss. It's because it's eating the keratin in the hair. The patient, of course, is going to want this to be treated like that, but treatment's going to take about 2 to 3 weeks because we have to get through this layer of fungus to make sure that we treat it, but we also have to make sure we don't hurt his own, cells in the process. So we have to make sure we're not giving him too high or too toxic of a level. A lot of our fungal infections out there are considered to be opportunistic pathogens. And again, that means that they're ones that usually are harmless unless they get into the wrong location. Now, again, a lot of times they can be in the wrong location. Another thing that can happen is they can be with a host that doesn't have a good immune system. So that can make them also opportunistic. If I don't have an immune system keeping me in check, then I'm going to start growing like gangbusters. Some of the infections that we worry about are, for example, things like Cryptococcus. Cryptococcus is an organism that can cause lung infections, and it's something that we see in immunocompromised patients. So in other words, if I got exposed to Cryptococcus, then my body, which is normally healthy and has a normally healthy immune system, would easily get rid of it and I'd be fine. But if I had somebody who was immunocompromised, who had a lower immune system, they wouldn't be able to clear the infection. They would end up with a really nasty lung infection. So again, a lot of fungal infections, it really seems to depend on what the person's body is like, what their genetics are, as to how much they're going to get ravaged by a fungal infection. In other words, it's hard to tell. We just don't know what people are going to get exposed to. Okay, I know that we have about 5 more minutes, but I'm going to go ahead and stop here, and we'll pick up with our protistas next time. Does that sound like a good deal? I'm seeing people are looking a little tired. I hate to drag people along. So we'll go ahead and stop here. We'll pick up with our Phylloprotista next time, and that way you have time to kind of review for your lab quiz and take a little break and wake up.